Hello everyone, my name is Louison Fresnais and I'm a third year PhD student working in collaboration between INRAE and L'Oréal. I would like to thank ECCB committee members for giving me the opportunity to present my work today, which will be about condition-specific modeling and network topological analysis to improve the understanding of metabolic mechanism of actions of chemicals in the context of systemic toxicity. So first, a little bit of context. Systemic toxicity refers to the toxic effects caused by the entry of a chemical substance into the bloodstream. And several initiatives to develop in vitro and in silico approaches have been launched, especially under the context of two consortiums. So the first one, Cosmetics Europe, and the second one, the ICCS, so International Collaboration on Cosmetic Safety. And they mostly developed methods that are based on differential gene expression analysis and gene set enrichment to determine potentially impacted pathways, therefore not directly representing the cellular metabolism. One thing to know is that systemic toxicity, among other types of toxicity, must be evaluated without animal testing before market approval. And systemic toxicity can also be studied through hepatotoxicity. So in this presentation, we are going to talk a little bit about amiodarone, which is an antiarrhythmic that is known to induce hepatotoxicity, but also pulmonary toxicity and thyroid disease. It is known that chemicals can induce metabolic changes, leading to metabolic diseases such as obesity, type 2 diabetes, and non-alcoholic fatty liver disease. So the idea is to use gene expression data obtained from cells exposed to chemicals and also genome scale metabolic networks to develop an in silico approach to better study cellular metabolic impact from gene expression data. Genome scale metabolic networks are composed of reactions and metabolites linked together by biochemical rules. So for instance, if I take the D-glucose here that is consumed by a reaction that is catalyzed by the exokinase to produce the D-glucose 6-phosphate, which in turn is going to be consumed by another enzyme that is going to produce the D-fructose 6-phosphate. We can apply this method to the large knowledge of human metabolism or any other species metabolism to construct genome scale metabolic networks such as Recon 2.2, which is constituted of more than 7,000 reactions, 5,000 metabolites, and more than 1,000 metabolic genes. So we developed a new workflow to study the metabolic impact of chemicals. Our workflow can be divided in two steps. The first step is a constraint-based modeling step, which aim at using gene expression data, for instance, in two conditions, to extract differentially activated reactions. And the second step, which is linked to the result of the first one, is a graph-based analysis step, which aims at performing clustering and subnetwork extraction to visualize the differentially activated reactions that have been predicted before. So yeah, the idea is to extract this kind of small readable subnetworks. So for the first step of the workflow, as I said, we are going to use chemical exposure transcriptomic data and the initial metabolic network, which is Recon 2.2. The first task that we have to perform is integrating gene expression data to the metabolic network. To do so, we use gene-protein reaction rules. And the idea is that, for example, if gene A is highly expressed here, then the enzyme A that is catalyzing reaction 1 is going to be also highly expressed. Therefore, the reaction 1 is going to be considered as highly active according to gene expression data. And we can do the same thing for more complex gene-protein reaction rules. For instance, if gene C and gene D are both highly expressed genes, then the associated enzymes are going to be considered as highly expressed 2, and the reaction 4 that is catalyzed by this enzyme is going to be considered as active according to gene expression data. Once we perform this step, we used Dexome for the condition-specific modeling parts, and Dexome will find a set of metabolic networks maximizing adequation between transcriptomic data and network topology. So Dexome is going to build the mixed integral linear programming problem, which is under constraint because we don't have enough biological constraints compared to the metabolic network complexity, and therefore we cannot find only one solution, but instead we are going to find a large set of possible optimal solutions. That's what we did, and for each condition we obtained around 10,000 solutions. So we have 10,000 condition-specific metabolic networks for the control condition, and 10,000 condition-specific metabolic network for the treated condition. And now the idea is that how can we extract knowledge from these thousands of condition-specific metabolic networks? So to do so, we perform the statistical analysis, which is based on activation frequencies. And for each reaction in the network, we computed a metric that is comparing the activation frequency of the reaction in both conditions, so in control and treatment. And if the R2, so if our metric absolute value is above 0.2, then the reaction is considered as differentially activated, meaning that this reaction is probably perturbed by the effect of the chemical. So once we extracted differentially activated reactions, we can visualize these reactions on the genome scale metabolic network. So that's what we did for our use case. So for amiodarone, we identified 56 differentially activated reactions on a total of more than 7,000 reactions in Recon 2.2. So we can roughly visualize these differentially active reactions on the complete genome scale metabolic networks with here the up-activated reactions after exposure to amiodarone in red, and the less activated reactions in green on this figure. But we cannot really infer metabolic impact from these visualizations due to the large amount of information not specific to the predicted metabolic impact. So the idea is to go from these complex GSMNs to human readable subnetworks that are specific of the metabolic impact of the chemical. So starting from the visualization that we've seen before, we are going to extract readable subnetworks with the same information, but with less not metabolic impact specific informations on the figure. So the idea is here to extract subnetworks that are interactive, focused on perturbed reactions, and allowing the analysis of the metabolic impact. To do so, we computed clusters of close reactions in the network using the metabolic distance. So for instance, we can map our differentially activated reactions on the initial metabolic network, which is Recon 2.2. For instance, let's say that we have 6 DARs, so R1, R2, R3, R4, R5, and R6. And we want to obtain, in fact, the metabolic distance matrix of these DARs. So the idea is that we are going to find the shortest path between all pairs of reactions with a shortest path algorithm. So for instance, between R1 and R2 we have a distance of 1. Between R1 and R3 we have a distance of 6. So 1, 2, 3, 4, 5, 6. And we are going to do that for all the pairs of reactions in the metabolic network. And at the end, we are going to obtain a distance matrix of all the metabolic distance between our pairs of reactions of interest. Once we have this distance matrix, we can apply classical hierarchical methods such as the hierarchical clustering on the metabolic distance matrix. So once we have identified clusters of differentially activated reactions, we want to extract a subnetwork for each of the identified clusters. So we are going to need the initial metabolic network, obviously, and the list of differentially activated reactions for the cluster that we want to study. And the next step is to compute the shortest path between all the nodes of the initial metabolic network in order to build what is called the metric closure graph, which is basically a graph with weighted edges, and the weight is the distance between two nodes. So once we have this metric closure graph, we can compute the minimal spanning tree approximation, and the idea is to find the minimal connected subnetwork spanning our nodes of interest. So on this example, we have our 4 reactions of interest, so R3, R4, R5, and R6, and another reaction that has been added for the connectivity. So now we can perform this graph-based analysis approach on our use case. As a reminder, we were able to roughly visualize differentially activated reactions, but not able to understand the link between perturbed reactions. And now with the subnetwork extractions and the interactive visualization from MetExplore-Vis, we were able to focus on each subnetwork and understand links between perturbed reactions, visualize associated pathways, and also visualize which metabolic functionalities are impacted. So to conclude and give you a few perspectives, we developed a new semi-automated system biology approach complementary to existing new alternative methods to better understand the metabolic impact of chemicals based on gene expression data. And we developed an approach able to extract mechanistic information from large sets of enumerated condition-specific metabolic networks. We also implemented a graph-based analysis approach based on metabolic distance and subnetwork extraction to visualize the metabolic impact of chemicals. And the subnetwork analysis and visualization features are key to see how these differentially activated reactions are connected, and also to facilitate the biological interpretation, therefore the safety assessments by toxicologists. We think that gene expression integration is an important step that we want to optimize, and also simplified visualizations could be of interest to compare the metabolic impact of large set of chemicals, because we have seen that they can be a little bit complex to analyze for our toxicologist colleagues, for instance. And finally, a last slide to thank people who collaborated with me on this work. I would like to thank Fabien Jourdan, Nathalie Poupin, Clément Fresnais, Jean-Clément Gallardo, and Maximilien Stingel from my INRAE team. Also, I would like to thank people from my L'Oréal team, and especially Olivier Perrin, Anne Rioux, Romain Graal, Bathilde Ambroise, and Alban Hott. And I would like to also thank Bernard Fromanti for his expertise in hepatotoxicity. This work has been published in bioRxiv and is currently under the reviewing process for another journal. And I would like to thank you for your attention.